The title of my talk is Systems Paleobiology, which is actually titled with an admiring wink toward one of the most rapidly expanding fields of research in the life sciences, which is systems biology. And as some of you may know, systems biology in a sense reflects the maturation of molecular biology from an initial focus on single genes and their products to the realization that the structure and function of organisms is really determined by the interactions of multiple genes and their products. And in a sense, the earth sciences have gone down a parallel track of increasing integration over the last 20 years, and that has certainly affected my home discipline of paleontology. So in a sense, we now understand in a better than we ever did before, that the narrative history of life that's given by fossils supplemented by phylogeny took place in the context of a dynamic environmental history. And what I'd like to do today is make an argument of how we might best understand the history of life in the context of Earth's environmental history. So let's see if this is... Now let me begin again with simply two panels of history, beginning in a place that all of you know, that is with the publication in 1788 of James Hutton's theory of the Earth. Most of you know, and Hutton is commonly regarded as the father of geology because of his insistence that the rock record could be interpreted in terms of processes that we can see today. But you might not know, unless you've actually taken the time to read the theory, that Hutton's motivation was explicitly biological. Like most late 18th century naturalists, Hutton was well aware of the goodness of fit between organisms and the environments they inhabit. So he looked at the hills and mountainsides of Scotland and saw that the trees and shrubs that lived there were well designed for that particular habitat. And he looked in the Firth of Forth and could see that the sea anemones and clams and algae there were also well designed for life in that environment. But what Hutton noticed that none of his contemporaries did was that those hillsides on which those trees and shrubs lived were constantly being worn down by erosion and through time would disappear. And he also knew that the products of that erosion would be washed down rivers and would accumulate in the bays, silting in the bays and then destroying the habitat where the clams and sea anemones lived. And so the dilemma for Hutton was how could life persist for long intervals of time in the face of constantly deteriorating environments. And as many of you know, his solution, which was a brilliant one, was to suggest that the environmental continuity was in fact dynamic, that an individual mountain would disappear, but some process, and he was relying on heat, would uplift other areas to form new mountains, so that in one way or another, there would always be mountains where these shrubs and plants could live. There would always be bays where the marine organisms could live. Now, about 50 years later, a little more than that, Charles Darwin sort of stood this logic on his head by demonstrating that populations were also dynamic and that life was no more constant than environments were. And if you then take these two ideas, one is that populations are dynamic and the other is that environments are dynamic, it raises the possibility that there could actually be significant interactions between evolving life and shifting environments through time. And that's really what I want to focus on today. So the nature of the argument is that paleontological data, increasingly supplemented by phylogeny, provide insights into the history of life, that in particular geochemical data from sedimentary rocks provide insights into environmental development through time, and the crux of the argument is that physiology specifically provides the conceptual bridge between the two. And what I really want to do is argue in favor of this way of thinking about the world rather than focusing on one particular aspect of Earth history. And I'm going to try and illustrate this argument with, at the outset, three vignettes. One showing how physiological insights can help us understand the time and space distribution of a single taxon then look at how physiology can help us understand biological reactions to short-term environmental perturbations, and finish up by making an argument that physiology also helps us to understand how biology co-evolves with long-term directional changes in the Earth system. Now, it's one thing to say that physiology provides this conceptual bridge. It's another thing to know how we can actually make valid physiological inferences from the fossil record. And I'll illustrate that just by thinking for a moment about this Jurassic oyster. One thing we can never know about this clam was its precise basal metabolic rate or its thermal norm of reaction. That's just not something that's going to be preserved in the geologic record. But we can know in a comparative sense that whatever the metabolic rate of this clam was, it was significantly higher than the metabolic rate of brachiopods in the same formation. And therefore, they might respond differently to episodes of environmental change. We can also make strong inference about processes where that are widely conserved within a group of related organisms, and therefore can be preserved, inferred from preserved morphology. So, for example, we can say a great deal about the process of biomineralization in this clam simply by knowing that it's an oyster and knowing how the range of variation in biomineralization in related organisms. We can know a great deal about its patterns of gas exchange, even though its gills aren't preserved. We can know a great deal about feeding mode. And I will point out that we know from the study of comparative biology that in almost any clade there's going to be exceptions to the rule. So there will be some species that don't fit the overall sort of phylogenetic expectations about physiology. But what that really means is that we have to consider these inferences in a statistical sense. And that's, if anything, what the fossil record provides us. It says that 88% of the clam genera at this point disappeared, or something like this. Okay, now let me start out by trying to get rid of this little sign. I'm not sure it's going to work. Anyway, you'll have to bear with it then. Let me start out with a plant called Medullosa. This was one of the major floodplain dominants in the ever-wet environments of low-latitude carboniferous coal swamp environments. It's been known for over 100 years that this was an unusual plant in that it had very large leaf systems supported by a very slender stem. And paleobotanists have also known, although I won't go into the details, that this plant had a highly unusual anatomy in terms of the plants that are with us today. And included in that anatomy was very large conduits for water flow. You can see here at the same scale, and it's a 100 micron bar, we have one single tracheid of Medullosa with the comparable conducting cell of a modern pine. Now, one of the reasons to start out by talking about plants rather than animals is that some of the most important aspects of physiological function in plants, namely the balancing of water loss with CO2 gain during photosynthesis, are explicitly biophysical in nature and therefore can actually be, in a sense, quantitatively reconstructed from the preserved anatomy of the plant. So if we want to reconstruct fluid flow patterns in this plant that's been extinct for 300 million years, we can actually go to measurements and models that have been based on living vascular plants, and I won't run through all the equations. The point here is to show that there are ways of making quantitative estimates of fluid flow in the conducting cells, this specific KSC, or specific conduction of this fossil plant. And the important thing is that in all of these equations, every term in the equation is either a constant, it's a variable of known environmental variation, the viscosity of water, for example, or it can be directly inferred from the anatomy of the fossil plant. So we can actually get quantitative estimates of fluid flow based on that preserved anatomy. And what you see here is modeling of that fluid flow on the left-hand side of extinct Medulosa. Here is using the same model for modern pines, and this is, again, a late Paleozoic plant called Chordites, which was basically a stem group conifer. And what you can see is that Medulosa was capable of conducting water at much higher rates than either pines or their extinct sister groups. And notice in particular that there's actually a difference in scale here. So this thing is really able to pump water at very high rates. And in fact, it's interesting, if you look at the conduit-specific conductivity of modern seed plants, you'll find that there's this, bifurcation between gymnosperms, which conduct water at low rates, and flowering plants or angiosperms whose vessels are able to conduct water at very high rates. And if you look at a textbook, it'll commonly tell you that one of the reasons for angiosperm success in evolution was this high rate of conduction. But what's interesting, if you look at plants that were alive within the first 50 million years of the evolution of seed plants, they actually had a much greater range of conductivities than we see in the modern biota, and Medulosa, in fact, conducted water at rates that are comparable to those of some angiosperms today. But that came at a cost. And if any of you were able to see Evan Anderson's poster yesterday, or you can see it later on today, you'll know that during the day when basically evaporation from leaf surfaces actually produces a tension, if you will, in the water column within the plant, generally spoken in terms of negative pressure. And that's why one of the reasons I loved Evan's poster is it's probably the only other person in this room who would ever use a crazy term like negative pressure. The important point of this diagram is that if you look at the thickness to span ratio of the conducting cells, at many values of this, you are actually safe, and the conducting cell is not particularly vulnerable to implosion, or equally bad to the development of air bubbles that would occlude flow. And both modern pines and extinct Chordites were very safe in the sense that their vascular system was not likely to be compromised by this. This is one of the reasons, by the way, that you see so many conifers in Norway. On the other hand, Medulosa was skating on the edge of disaster all the time. Basically, at thickness to span ratios down here, there's actually a very high probability of implosion or cavitation at really pretty moderate negative pressures. This is the kind of thing that you might see in Boston on a warm July afternoon. So this suggests that although this plant was conducting at high rates, it might have needed ways of either providing redundant routes for water so that the occlusion of one route wouldn't be fatal to the plant, and or it would be able to actually resupply the vascular system from surrounding living cells. Okay, to finish this up then, the physiological connection then is we can actually show that this ancient group of land plants, which was widespread in tropical ever-wet environments, but which never particularly invaded higher latitudes and which went completely extinct as the coal swamp environments disappeared toward the end of the Paleozoic, it conducted water much like at rates comparable to those of modern angiosperms, but would have been quite vulnerable to damage induced by water stress. And in fact, if we look at the total anatomy of this plant, I won't go into it in detail, its anatomy really does seem to be optimized for essentially circumventing the problems of these water stress issues. And this helps to explain why this plant was distributed the way it was. It was found in these ever-wet environments because it simply couldn't survive drought or prolonged freezing, and therefore was physiologically unable to invade these other environments successfully. And it also helps us to explain, as these warm, ever-wet environments disappeared toward the end of the Paleozoic, Medulosa disappeared with them. Okay, let's shift now and look at an extinction boundary, and we might as well talk about the big one at the end of the Permian. Most of you know, and I hope at least some of you saw the good poster on this yesterday, that after a prolonged interval of more or less constant diversity of life in the oceans, at the end of the Permian, we basically lose the great majority of the genera. Perhaps as many as 90% of the species in Lake Permian marine rocks are not found in the Triassic. This is simply at Maishan in China, where you see dramatically that these limestones of latest Permian age are just full of all of the life that characterizes late Paleozoic marine systems, full of brachiopods, foraminifera, bryozoa, things like this. And at a point into which you could insert a knife blade, all of that disappears. And as we sort of repopulate the oceans with skeletal fauna going up into the Triassic, it's largely different taxa than those that were abundant here. So a major mass extinction. And again, as you could have seen yesterday in the poster, there are two circumstances that seem to have played a major role in the physical perturbations that drove this extinction. I'd stop short of saying there's consensus among earth scientists on this, but many people would argue that because of the tectonic circumstances of the Earth at the end of the Permian, with this agglomeration of the continents into Pangaea, that a consequence of that is you actually have an ocean basin which is larger than a hemisphere in dimensions, and with little or no glacial ice at the poles, that there would have been somewhat sluggish physical circulation, promoting oxygen depletion in subsurface water masses. At the same time, or at the moment of extinction, within our ability to say this, there is massive volcanism in Siberia, the Siberian traps. Again, the estimates of the volume vary from 1 or 2 million cubic kilometers to as much as 7 million. If you suggest that the amount of CO2 emitted from these volcanoes would be comparable to the rates of CO2 emission from, say, Hawaii shield volcanoes, you would come up with something on the order of 10 to 1,000 times the amount of CO2 in the late Permian atmosphere was injected through this event. And that probably is a low estimate. We know today that something like 12% of all the CO2 emitted from all the volcanoes and ridge systems on Earth comes from Etna and Vesuvius, because they actually plumb through carbonate platforms. And indeed, these late Permian or end Permian flood basalts actually plumb to several thousand meters of Proterozoic carbonates and were extruded in many places onto coal swamps, which would give you thermogenic methane as well as CO2. That being the case, there's no shortage of proposed killers in here. Carbon dioxide itself could be an agent of mortality and extinction in at least 3 physiologically distinct ways. You're all familiar with ocean acidification, a current issue when you actually raise CO2 at rates that are faster than the Earth can buffer them. pH goes down, the saturation state of seawater with respect to carbonate minerals goes down, and it becomes more challenging for skeletal organisms, or carbonate secreting organisms, to make their skeletons. At the same time, there's a fairly large literature on the direct physiological effects of high CO2 levels in bodily fluids called hypercapnia. And then, of course, injecting a lot of CO2 into the atmosphere would be an agent of global warming, which could add yet another source of mortality. So carbon dioxide is a sort of multi-purpose killer here, which is something that excites paleontologists a great deal. Now, also temperature itself, through its influence on the function of organisms, both land and the sea, could actually be a killer, and you would make predictions about the direct effects of temperature based on physiology times a geographic setting. It has been noted empirically that there is an expansion of anoxic subsurface water masses, which could, of course, cause asphyxiation as well as contributing to CO2. And it's also been suggested that in these anoxic areas, there would be a buildup of hydrogen sulfide causing sulfide toxicity. The point I want to make here is that each of the kill mechanisms that are implied by these physical disturbances carries with it an explicit set of predictions for what organisms would be more likely to be affected and what organisms would be less likely to be affected. And I'll only talk about CO2 because of all of the extinction mechanisms that have ever been proposed for the Permian. The only one that actually correctly predicts what actually went extinct and what survived are the physiological effects of CO2. So let me just quickly show a few slides. Many years ago, we actually suggested this, and what we simply did was, first of all, spent several months just reading physiological literature on CO2 and found that based on patterns of skeletal biomineralization, one would expect some organisms to be more vulnerable than others. Based on both basal and exercise metabolic rate, one could make predictions about vulnerability. Anatomical complexity makes predictions, and actually the physical setting of the organism, whether it's living on the surface of the sediment or within it, makes predictions. And so what we did simply was took the Permian and Triassic marine biota and divided it into two bins based on inferable features, physiological features. And what you'll see that those organisms that we predicted to be sensitive to CO2 really got smashed. And while there are extinctions among those that were predicted to be more tolerant, they had much, much lower rates of extinction. Now, one of the things that's happened since we did that initial work is that both societal and scientific concern about ocean acidification has led to a remarkable expansion of physiological research on marine organisms. And one of the leaders of that research, Hans Otto Portner in Bremerhaven, actually said, why are you guys being complicated here at the Permian? Why don't you just look at individual factors and see whether the physiological predictions of, say, skeletonization biology might be sufficient to explain your pattern? And that was actually very good advice. So when we came back to this, we simply made 3 bins based on skeletonization. On this bin are organisms that have a large commitment to calcium carbonate skeletons, but only a limited physiological ability to modulate the composition of the fluids from which they make those skeletons. Corals would fit in here, sponges, brachiopods, echinoderms. At the other end are organisms that have either very little commitment to a carbonate skeleton, making only small spicules, or actually have skeletons made of other materials. And in the middle, mostly mollusks and arthropods are things that have a commitment to a carbonate skeleton, but have at least some ability to pump ions across membranes in ways that will actually allow them to modulate the chemical composition of their fluids. And you can see that just by making these simple bins, you see that anatomy was really destiny at the end of the Permian. If you were a genus of hypercalcifiers at the end of the Permian, as a genus, you had about an 86% probability of not making it into the Triassic. If you didn't use calcium carbonate in your skeleton, you only had a 5% probability of extinction. That is, the organisms in this bin actually don't notice the end Permian extinction. They don't have extinction rates any higher than background. And interestingly, for this intermediate bin, which shows intermediate levels of genus-level extinction, if you then go back and subdivide this using the other factors that would, we think, either promote survival or vulnerability, those that you would judge on that basis to be more vulnerable have extinction rates twice those of the ones we'd predict to be less vulnerable. So again, pretty much every kind of test that we and others have been able to throw at the end Permian suggests that the physiological effects of rapid and large-scale elevation of CO2 are likely to have played an important role in the extinction. Okay, the physiological connection then is simply, there was a lot of argument for a decade of, was the extinction caused by high CO2, expansion of anoxia, sulfide, temperature? And the important point to say is that in an integrated Earth system, you inject a lot of CO2 into the atmosphere, and you get all of these. They are not independent actors in the Earth system, and so that all of them would have been in play at the end of the Permian, and all of them could have been, and probably were, agents of mortality. And in fact, we know from modern physiological work that they are synergistic in their effects. That is, the effects of CO2 are made larger when the temperature is warmer or when oxygen levels are decreased. Having said that, rapid CO2 increase seems to have shaped the pattern, the taxonomic pattern of selectivity, which is, of course, what is going to determine those organisms that are around to repopulate ecosystems in the in the Triassic. This is just a recent paper from Justin Rees showing that as people are looking at the effects of decreasing saturation state on calcification rates in a wide variety of marine organisms, there are actually a wide variety of responses. And what's interesting to me is that the difference in response to changing omega among marine organisms today pretty much mirrors the vulnerability to extinction at the end of the Permian. And I'll close this section by just reading a quote from Frank Meltzer at Kiel, who summarized a decade of research on ocean acidification recently by saying all more tolerant taxa are characterized by high specific metabolic rates and high levels of mobility and activity. And that would actually be a very accurate summary of who survives the end Permian crisis. Okay, third and last geological example then, and that is looking at long-term directional changes in the Earth system. And as many of you are aware, there's probably no stronger long-term environmental vector than oxygen. The triptych that I show here, is one that Ariel Anbar and I published about a decade ago, based on really some pioneering work by Don Canfield in Denmark. And what it does basically is breaks down the whole of recorded Earth history into 3 large chapters. At the time we wrote this, and certainly today, there's agreement that in the Archean, before 2.4 billion years ago, there was little or no oxygen in the atmosphere and water column, although ferrous iron would have been relatively abundant. And I would just say parenthetically, I really believe that on the Archean Earth, the carbon cycle and iron cycle were closely intertwined. Neither was there much argument about this panel, that is, during the Phanerozoic, at most times and in most places, the water column of the oceans has been fully oxygenated. We know there are exceptions to that, but they're taken as exceptional, and things like oceanic anoxic events get a great deal of interest when these transient events happen. But the real burden of the argument is that these 2 worlds were separated by a long-lasting intermediate redox state of the Earth system in which there was a little bit of oxygen in the atmosphere and surface ocean, maybe a couple % of today's levels, but that when you got into the oxygen minimum zone, that there was a strong statistical tendency toward anoxia. In some cases it's sulfitic, but we now know that in some cases there was enrichment in ferrous iron rather than sulfide. We know less about the deep ocean. One would guess that it would have been kind of a marble cake mixture of dysoxic water masses, anoxic but ferruginous, and anoxic but sulfitic water masses. The important thing is that for those organisms living in the surface ocean, it's what's going on just beneath them that probably has the strongest effect on biology. Now, how do we know that? Again, I won't go into it in detail. I will be happy to talk to anybody. For individual basins, we can look at something called iron speciation chemistry, which is simply the forms in which iron is found in mud rocks. And what we find is that through that intermediate interval of the Earth, when you look at mudstones deposited in shallow shelf environments, their iron speciation chemistry is much like that of muds being deposited beneath oxic water columns today, but that for several hundred million years, whenever you go and sample muds formed at maximum flooding in basins, and therefore their deepest water is represented, they basically show signs of accumulating beneath an anoxic water column. One can also look at the abundance and isotopic composition of molybdenum, a redox-sensitive element, which will, in a sense, give a more integrated view of how the ocean is working. Those also suggest that there was much, much more widespread sulfide-containing water bodies in Proterozoic oceans than there have been since that time. Jochen Brox, when he was a postdoc in our lab some years ago, actually looked at the organic geochemistry of some of the rocks that formed during this interval and found that, among other things, there are biomarker molecular molecules synthesized only by anoxygenic photosynthetic bacteria. In plain language, that means these are photosynthetic bacteria that are getting their electrons from sulfide rather than water. And what that tells us is that at this time and place, you had to have anoxic water masses within the photic zone. And there's widespread agreement that this was a long-lasting, low-oxygen environment. Now, as a paleontologist, that's interesting because these are the oceans within which the modern eukaryotic cell comes to exist. Both fossils and molecular clocks indicate that the last common ancestor of living eukaryotes probably lived 16 to 1800 million years ago. We see fossils that I think are unambiguously eukaryotic in these middle Proterozoic rocks, but they're living in a world that is very different from any world that we understand well. Certainly, anything living in these surface waters would have been challenged by episodic mixing of anoxic and sulfidic waters into the surface. Some of you will know that sulfide in particular inhibits mitochondrial activity and therefore is broadly toxic to eukaryotic organisms. Also, if you try to understand the nitrogen chemistry of waters in a water column that has a redox profile like this, there's reason to believe that fixed nitrogen would have been in short supply in surface waters, and therefore there would have been an advantage to primary producers capable of nitrogen fixation. No eukaryotic alga can fix nitrogen. Most bacterial or cyanobacterial photosynthesizing organisms can. It turns out that if you actually look at the fossil record from this interval, the observations actually fit these predictions. Although eukaryotes existed throughout this period of Earth's middle age, there's limited diversity and limited ecological amplitude. That means that whenever you're most likely to find fossils that appear to be eukaryotic hugging the coastline and not out on the shelves. Biomarkers also suggest that throughout this interval, even though eukaryotes existed, primary production was done mostly by bacteria. And curiously enough, one of the earliest places where we start to see eukaryotes coming to taxonomic and ecological dominance is actually in lakes, where there's independent evidence that they're becoming more fully oxic earlier than the oceans themselves. About 800 million years ago, this situation starts to change. We start seeing a fairly sizable increase in the diversity of eukaryotes, and I'll particularly point out these things found in the Grand Canyon in 750 to 800 million year old rocks, which are very similar and probably both ecologically and taxonomically related to things called testate amoebae that live today. And what's interesting is that these are our first evidence of eukaryotic cells that could actually prey on other cells of comparable size. They're not just picking little bacteria. They're able to eat their brothers, if you will. Interestingly, at the same time, we start seeing armor. Apparently there was some use at that time for eukaryotes to armor themselves against predators. And because we've just been working on this, let me show you this beautiful stuff. These are scales of the type that modern chrysophytes and other protists form, except these have been etched out of 800-million-year-old rocks in Canada. This is about a 10-micron scale. And it turns out these scales are now the most diverse record of eukaryotic life we have before the appearance of animals. And so for some ecological reason, these organisms are armoring themselves, and it's at least convenient that they're armoring themselves when we start to see eukaryotes that prey on other eukaryotes. One other predicted consequence of protistan predators is that some organisms will become multicellular to avoid predation. There have been some beautiful experiments in the biological literature where people took populations of single-celled algae, added predatory ciliate protists, and within something like 10 generations, the green algae are obligately multicellular because those organisms can't be eaten by the ciliates. It is thus at least interesting that molecular clocks now suggest that the last common ancestor of animals existed on the order of 750 to 800 million years old. If that's true, and there has to be some prehistory of animals, whether it's exactly this or not, we have some interesting explaining to do because we actually don't see macroscopic, that is, large-size visible animals in the fossil record until about 580 million years ago. And one has to ask, why would there be this long interval of evolution before we actually see animals? And the physiological best answer to that is that something was keeping animals small and unapparent and unlikely to fossilize, and oxygen is maybe the most interesting candidate there. So we can go back to the geochemical record and say, when did geochemical data suggest that we first start to see oceans with more fully oxidized or oxygenated water masses? And again, I won't go into the details, but arguments based on sulfur biogeochemistry, arguments based on both the abundance and isotopic composition of molybdenum, arguments based again on this iron chemistry through a number of late Proterozoic basins are now converging on the idea that oxygen levels began to reach the levels capable of supporting the physiology of large active bilaterian animals at about 580 to 560 million years ago. Interestingly, one might predict that as oxygen goes up, nitrate abundances should increase in the oceans, and eukaryotic organisms should become more important as primary producers. And again, both fossils and biomarkers suggest that that's the case. Only toward the end of the Proterozoic do we see especially green algae becoming important parts of the phytoplankton. So the punchline then is that once again, physiology may actually help us to link the biological and the environmental record, suggesting that although animals have a potentially fairly long prehistory, it is only during this interval of 580 to 560 million years that we accumulate enough oxygen to support the life of large motile predatory bilaterians. Okay, if I have just a few minutes, let me take this argument to another planet for just 5 minutes, and then I'll finish up. This is a lovely picture of the surface of Mars taken about 5 years ago by the NASA's MER or MER rover Spirit. And you might say, well, that doesn't look like a particularly biologically interesting landscape. And you're exactly right. I mean, this is a terrible place to think about any life we understand. As some of you know, the pressure and temperature of the Martian surface is such that liquid water isn't even stable in these environments. There's harmful radiation. There's all sorts of strong oxidants in the soil. I think it's very unlikely that there is life present on Mars today. However, there's a lot of interest in Mars as a biological planet because both geomorphology and sedimentary geochemistry tell us that, in fact, in the past, Mars did, at least episodically, have liquid water. And so the last example of the systems paleobiological approach I want to explore is just to say, how does it help us to constrain speculation about life on early Mars? It's not enough to say that water was once present on the Martian surface. We have to say something about the properties of that water and the persistence of that water in time. As some of you will know, the Opportunity rover that I've worked on for the last 10 years has found this wonderful sedimentary column, tens of meters thick in Meridiani Planum. This, I'm actually very proud of this. This is actually the first stratigraphic section ever measured on another planet. It's about 7 meters thick. It shows that these rocks that you see here are largely aeolian, but there's at least two types of evidence for water. One is if you look at the composition of the sand grains that are making up these sandstones, they are in no small part sulfates. And since the parent rocks for these are basalts, you have to have water-rock interactions to generate mostly magnesium sulfates, but also some iron and calcium sulfates. You actually see the kind of things you would see in comparable modern environments on Earth, where you have fluctuating water table with zones of recrystallization. And at the very top, you actually see evidence of types of ripple marks that are only likely to form in the presence of a liquid. So it looks like you transiently have something like a playa lake forming here. Now, what can we actually say about that water? And I just want to say a word or two about water activity, which is in some ways a fancy way of talking about salinity. And I'll call your attention to Samuel Taylor Coleridge's poem, The Rime of the Ancient Mariner. Some of you will remember this poem. Here's this poor mariner. He's in a little boat. For hundreds of kilometers in any direction, there's nothing but water, and he's dying of thirst. The question is, why is the ancient mariner dying of thirst? Coleridge tells you it's because he killed an albatross, but physiologists know it's because humans cannot tolerate water for very long at the salinity of seawater. And it turns out that every organism that we know of on Earth has limits to the water activity that it can actually survive. So in water activity space, 1.0 would be distilled water. Seawater has a water activity of about 0.98. So most of the life that we know is up here, lots of organisms. As water activity goes down to the level of orange juice concentrate, for example, only a relative handful of bacteria can survive. You go down to saturated halite solutions at a water activity of about 0.74. A very small handful of archaea and fungi can survive. And the world record right now for surviving and actually growing at low water activity is a fungus called Xeromyces bisporus. Honey down here is kind of interesting because you've all actually done an astrobiological experiment in your own homes where you open up a jar of honey, take out a little bit, put it back on the shelf, and then forget about it for a decade. The question is, does it spoil? The answer is no, it doesn't, because honey's water activity is lower than that known to support any life on Earth. You put water on that honey and it'll spoil in days. And I should say that the reason it's not by accident that this kind of graph has things like orange juice concentrate and honey on it. It's because the whole science of food preservation is based on excluding microorganisms under conditions of reduced water activity. So the important thing is that by using some simple thermodynamic calculations, you can actually back out the water activity of a fluid on the basis of knowing the minerals that are precipitating from that fluid. So if you take a jar of seawater, start evaporating it, the concentration of the different species in that seawater will increase, but only for so long, and then you'll start precipitating minerals from it. So gypsum comes out first, then halite, then Epsom salts, and finally things like magnesium chlorides like bischofite. The important thing here is that you can display that same chemistry in terms of water activity, and you will know that gypsum comes out at a relatively high water activity, halite somewhat later, epsomite somewhat later, etc. So the point is, if you can make a reasonable estimate of the composition of the dilute fluid from which these were eventually precipitated, you can back out the water activity. And my former postdoc, Nick Tosca, now at the University of St. Andrews, did this for Mars, making, I think, some very reasonable and conservative estimates of the Martian situation. And this is the water activity. And you'll notice that the dominant salts at Meridiani Planum, where I showed you that stratigraphic section, are magnesium sulfates. We've used epsomite in the modeling. I should say we don't know the hydration state of these magnesium sulfates, but every other mineral we could have used would make this worse. And you'll see that by the time Epsom salts come out on Mars, the water activity would basically kill all but a handful of organisms on Earth. We also have evidence for halite precipitation as a late stage evaporite in many places in Mars, and halite on Mars has a water activity that no known organism on Earth can tolerate. Now, so the question always comes out, what if it's an alien biology? And I think that all that tells us is that games without rules have lots of different possible outcomes. What we can really say is that no life that we understand would do very well in these waters as they evolved on the surface of Mars. So we can actually take this physiological relationship between organisms and environment and do the inverse problem and say, knowing something about the environmental history of Mars, can we constrain speculation about biology? And certainly both the kind of exercise I've shown you, also work that we and others have done that shows that rocks that have been exposed at the Martian surface for intervals as long as the Phanerozoic have essentially no aqueous alteration on them, suggesting they haven't seen much water. And another paper that Nick Tosca and I did, in 3 to 4 billion-year-old sedimentary rocks on Mars, you find all of these sort of juvenile precipitates, amorphous silica, things that have to be precipitated in the presence of water, but which actually on Earth are transformed into other minerals in the continued surface of continued presence of water. So the fact that you still have things like amorphous silica and jarosite in 4 billion-year-old rocks on Mars, again, tells you that these couldn't have seen much water. So that suggests that if there was a window for any kind of life that we understand on Mars, it must have been very short and concentrated in the earliest part of recorded Martian history. I will point out that we have one little glimpse of that earlier interval of Martian history just published last week in Science by the MER team, and that is at the base of this whole thick succession of sulfates, they basically peter out against an older elevated crater margin, and in the earliest sediments of that we actually find gypsum veins. And that's the first evidence that there was, at least for some interval of time, water of moderate temperature and salinity. So let's just finish it up then. The whole point is that life is not simply an evolving system on top of an inert planet, that on all time and space scales, life and the environment are interacting in ways that affect both. So we know that in the Phanerozoic and in the Precambrian as well, you have shifting continents, you have changing climates, you have changing atmospheric composition. And of course, I point out that through human agencies, we are probably changing the environment at rates that are as fast as anything that we've seen since the Permian extinctions. And so therefore, both to understand the history of life and increasingly to understand the future of life over the coming centuries, I think we have to develop this calculus of using physiology to understand the relationships of life and environment. Thank you very much.